Okay, thank you for the nice introduction. I feel a bit bad because it should have been Adriana Lopez dos Santos who would have presented this talk. She's ECODIM alumni also, and she has been also a TA. She came several times to, but unfortunately, she's stuck in Brazil. So initially I was thinking of presenting your talk actually, but then I said, okay, I'm going to go slightly differently. So the title I changed. If you look at the title like 2 days ago, and then I prepare and say, okay, I'm going to take a broader perspective. So what I'm going to talk today is about the biogeography of marine phytoplankton, with an emphasis on polar region. Initially, the talk was going to talk only on polar region and on the dynamics of the under ice bloom. I will talk about this, don't worry, but I will take a broader perspective. Oh, let me start my clock. So I have 1 hour like everyone, or? Yeah? Or I'm already done. Okay, so I'm going to you presented Rosco. I'm going to give you some idea where Rosco is, so then during the course you know where I am. I'm going to talk very briefly about marine plankton, metabarcoding, and the database we have been developing in the last 3 years. And finally, I will really jump into the topic of how this database can be very useful to look at broad distribution of marine phytoplankton. So this is going to be an introductory course on phytoplankton, but most of them work with bacteria or viruses. So I think that might be useful. But before Rosco, so Rosco is, oops, I know, there is no pointer. Yeah, I found, sorry. Okay, so Rosco is here, UK is here. This is the English channel, so, and Paris is here. It's quite far from Paris, actually, 600 kilometers from Paris. So it's one of the, so, okay, we have a problem with this. It's not very good. Rosco is one of the oldest marine biological stations in the world. I think it's number 3 in terms of creation date. And this year we were doing the 150 years of the station. It was founded by this Professor Lacaze-Dutier in 1872. And it has a staff about 350 people, and about 1,000 to 2,000 students go every year. I'm part of this team called EcoMap for Ecology of Marine Plankton, which is about 10 scientists, 40 people all together, and we work on many research topics, viruses, bacteria, cyanobacteria, symbiosis, parasitism, all things that you will hear either today or during this class. Actually, I'm retired now, and I spent the last 4 years, I'm still emeritus in this team, but I spent the last 4 years in Singapore at the Nanyang Technological University. This is going to be a pen, huh? Sorry, it's not a PowerPoint, it's a PDF, but it's not a reader for PDF, so it's not so good. The Rosco Cotter collection, so it's a very active collection of marine algae, probably the largest in the world now. We have about 5,000 strains that we distribute. So if you want to work, and I know some of you have worked on marine strains, it's a very, very good and useful resources. Okay, so marine plankton. So you have seen some kind of pictures like this from Matt, I guess. So marine plankton and phytoplankton is at the core of all the cycles in the ocean. It's of course in the surface layer. and it plays a critical role in many, many processes. So there is different types of plankton, of course. You have, and this is image, some of the image, this is from Tara actually that Matt mentioned. So you have zooplankton, phytoplankton, lot protists, and many more people now are working protists, of course, bacteria and viruses. But I'm going to focus on phytoplankton. And phytoplankton is very diverse, both in terms of shape, because you have cyanobacteria, diatom, dinoflagellate, green algae, and many more. But more importantly, in terms of and then in the phytoplankton also there is a very small phycoplankton, which is important. And phylogenetically, it's also very diverse. Basically, you can find phytoplankton in most of the branch of the eukaryotic tree. There are many questions to be answered about the role of phytoplankton, but one type of question I've been interested for a very, very long time actually is what is the distribution of the phytoplankton species or phytoplankton taxa? For example, in terms of latitude, of course, like polar versus temperate versus tropical, coastal versus pelagic, surface versus euphotic zone, and also in terms of seasonal cycle. And all these questions about the biogeography actually of the phytoplankton has relevance to how the ocean is going to react to global change. Because if you have change in the temperature, you are going to have some species which are going to move north. Even in the tropic also you can have some change and so forth. So they are important to understand the change of the community, and also maybe to have some indicator species that are going to tell us information about changes in the ocean. So before people were investigating these questions by microscopy. Okay, so you were taking samples, and then there are some large-scale studies that have been made available where you can understand better, for example, the distribution of specific taxa. For example, this is a species called Noctiluca that we have actually in Singapore. It does very nice bloom, it's fluorescent. And you can see, for example, here in this data set where it appears. And then you can try to understand what is the reason for this species to appear, and maybe use it as an indicator species. There are even a larger data set based on microscopy, for example, here. But still, first, it's very time-consuming to obtain this data, and also there is a lot of uncertainty based on the operator who is doing the analysis, because nobody can do so many analyses. So you have, so that's where I want to introduce metabarcoding that all of you have heard about, but I will just very briefly summarize the approach. So the idea is to take a water sample, to take a universal gene and amplify it, sequence it, compare it to reference database, and then we can have some information about the sample composition in terms of. So it's not fully, of course, quantitative. I will say it's quantitative in terms of relative quantitation. But still, the more data we get, oops, what's happening? Yeah. Huh? What's
Someone turn off unplug the computer. Oh, no?
No, I didn't touch anything. The computer is there. Can all come around and watch the computer, but it's not going to be. Ah, it's coming back.
Yeah.
So I have to
don't
do
know.
this if it happens
Yeah.
again.
And it's passing the
Huh?
working the passing the.
No, it's okay.
It's okay.
Well, it's not perfect, but it. I'm not talking. Okay. Oops. Strong enough. I think Jake, you throw something. There was like something that somewhere on the floor actually to make it more smooth, you know. So so I'm going to focus on eukaryotes. I'm not going to talk at all about prokaryotes, or bacteria and archaea. So for eukaryotes, we are using the gene that is used most is the 18S ribosomal gene. You can use also other genes. For example, people who are working on fungi, they use ITS because it's more resolutive. And in this gene, of course, you cannot, with the high throughput sequencing, you cannot sequence everything. So you choose a region. And for eukaryotes, usually is a V4 region, which is located here, which is about 380 nucleotides. So this varies between different species, of course. The advantage, it's very suitable for current Illumina technology of 2 times 250 or 2 times 300 base pairs, so 600 base pairs, so you have a good coverage. And there is a very large database of reference sequences because a lot of people were sequencing this region in the old time when we were doing clone libraries or for sequencing individual species. One of the problems is that some groups have large introns, so you may miss some groups, but I think by large, V4 is one of the best regions. There is another region called V9, which is at the end of the gene, but which has much less coverage in database. So then you get all your sequence. Typically, no, not this one. Okay. Oops. Sorry. Sorry. So you get typically, for example, you get, depending on whether you are rich or poor, you get 1 million sequence. If you are very rich, you get 100,000 or 10,000 if you are not so rich. Then you use some bioinformatics, and hopefully maybe during the end of the course we will talk about this. Actually, I will show you how to use this R package called Data2. So you make auto use basically, or it's called ASVs, and then you basically put them in a tree, or there are other methods. Yeah, good now. You assign them to a taxonomic group. Depending on the group, you can go down, of course, to the species level, and V4 is good even to go down to the species level. So really, you can go down to the species level for some groups, not for all, but for some. But for this, you need a good database, and that's where this database that has been developed over many years called PR2 is quite useful. For bacteria, there is another one called Silva, but Silva, if you are doing protists or eukaryotes, don't use Silva because it's bad, actually, it's not very well maintained. It's going to get better now because it's going to be part of a large culture collection called the DSMZ, where I think it's going to have a refresher. But so work with, so PR2 has about 200,000 reference sequence here, which are associated with 8 taxonomic ranks. So basically for each sequence you get all the different ranks here. It comes from many studies, either culture or clone libraries. And this database has also some metadata. So you know where the sequence is coming from, like in terms of whether it comes from a culture, whether it comes from oceanic environment, and so forth. So you can use this for signing your metabarcode. And basically, a metabarcode data set is quite simple. It's just a matrix where you have on the here you can have this as an Excel sheet. Each column is a different sample, and each line is a different sequence, ASV, or think it as a species. It's not more complex than this, but you can. And here it's just the number of sequence for each of these ASVs. So it's really no more than this, actually. Nothing really very, very fancy. But the whole bioinformatics is to get to this table from your original row sequences. Okay? So, and then after, and hopefully during the course, we may have a chance to do this. You can use some software to do some analysis. So you can do like multivariate analysis. Matt showed some multivariate analysis for viruses, but and I think Rodrigo will talk about this later. So I'm introducing everyone here. Yes. I hope you got. So you can, and we will, do this later. Anyway, so PR2 is a reference database. It's long sequence which have been characterized, for example, from culture. But also in the last 10 years, there has been an abundance of metabarcode studies. Okay, so one of the most famous that was already, I'm doing a lot of publicity for Matt here because he's talked about everything, from Tara. Okay, so Tara was, he did a lot of metagenomic sampling and analysis, but he did also metabarcoding analysis. And this is probably one of the most cited for marine or plankton paper by Colomban de Vargas in Science, I think, Nature, Science, whatever. I think it was Science, where they could analyze a community from different size fraction. Now I keep talking, even if you don't see the picture. For different size fraction in different oceanic basin, and they found very, very interesting facts about many diversity which was really underestimated before, and also there's been a flurry of patterns actually of papers using this data, because these data were put right away in the public domain, and there has been a lot of analysis done actually based on this data. So this proved to be a very, very useful data set. But this is not the only data set available. You have many other metabarcoding data sets. For example, you have Ocean Sampling Day, which was a study which sample coastal environments. Tara is mostly marine, open ocean, while OSD, Ocean Sampling Day, got samples from all over coastal places, mostly in Europe and US, but also a bit elsewhere. There is a very interesting study of protists in the gutters of Paris. So the people, they sample all the gutter, where you put the water in Paris, and then so all this data, the problem is that they are available in the paper, each paper, but usually it's very difficult to compare them because they are not in the same format. They've been using different software to do the analysis, different, for example, similarity level to build the OTUs. And metadata usually are very hard to find, not in an easy way. And people have been using, it's come faster and faster, I feel. It has like its program. Most of the people have been using these large-scale studies like Tara or Malaspina, while it's a bit, the resource is here, but nobody can use it really because the data are there. So the idea of what I call meta PR2, so PR2 for metabarcodes, is to reprocess all the raw data from the study. So we go in the literature, we find studies that are very interesting and covering a large environments that are not covered, for example, by Tara or OSD. And then we use this software called Data2 that I will introduce during the course to reprocess this data. We annotate the taxonomy with a single database because sometimes that's a problem. People are using different databases to annotate, so it's very difficult to compare, called PR2. And then we integrate also a lot of metadata. So all this has been leading to a database which is called MetaPR2. The paper was published last year. And the last version of the database has been released this winter in December. We have so now 59 data sets, which correspond to 6,000 samples. And altogether, this is more than 90,000 different metabarcodes. We have data, of course, we have Tara Ocean included. But it was completely reprocessed because the initial processing of Tara Ocean was done with another software, which has different parameters than Data2. Malaspina Ocean Sampling Day, a lot of Arctic data sets and so forth, and also some data from lakes, rivers, and soils. So all this is mostly, for the time being, unfortunately, we have mostly oceanic, but we still have some coastals and freshwater lakes, and even terrestrial, like soil samples and so forth. But still, there is many other data sets that can be added. To make this available, I mean, if you provide this as a simple file, it's going to be very hard for people to explore it. So we developed, using the R software, a web interface that you can access here, that allows you to do a lot of analysis on the different metabarcodes. For example, you can select for a given taxonomy group, okay, you are only interested, for example, in diatoms, then you can select here, okay, I just want to see the diatoms. I'm only interested in oceanic samples, so you can have a lot of selectors for different samples, and then you can visualize on maps the abundance of your taxa and the diversity of your taxa. And then you have many more functions. So you, for example, you have maps, which are very, very useful, of course, for many analyses. You can also compute alpha and beta diversity. More importantly, if you have your own data, you can query the database. Okay, let's say you are doing a study, you find a metabarcode that doesn't seem to correspond to anything. You can do a blast-like search on the database, and it will provide you a map of the location where this sequence has been found, actually. So that's okay. So far so good. Okay, so how we can use this now? So I'll show you 3 examples in terms of the biogeography of phytoplankton. I really like this. This is from Humboldt, right? And there's been this fantastic book about Humboldt. And I really think he is, I never read Humboldt in the text. I don't read German anyway. But I think this is really a founder of ecology in some way, with his analysis of the vegetation on the volcanoes of South America. Okay, so I'm going to take 3 examples. First, a very simple example of two new species of ice algae that we found and how we use this database, or it was a embryonic database at that time to show where they were really distributed. Then I'll show work to which I participated on diatoms isolated from Svalbard, and finally I will finish with under ice bloom, which is work ongoing with a team of Adriana at NTU in Singapore. So just to give you some very simple use of this kind of data, but still very useful, I participated to two cruises in the Canadian Arctic, one in the Beaufort Sea and the other one in Baffin Bay, and I will come back to Baffin Bay later. And we isolated these two Mantenilla species, one in Beaufort Sea and one in Baffin Bay. So we were not very imaginative. We call one Beauforty from the Beaufort Sea, and the other one Baffinensis from Baffin, Baffin Bay. And we sequence, of course, the 18S, and so this strains used to be in the Colta collection. Unfortunately, we lost one, the one from Baffin we lost, because these are not very easy to maintain. And then the map you are seeing here is a sequence which are similar to the Mantenilla sequence of these two species in this database, which was still, there were not that many points. There was Tara and I think OSD, not even Malaspina. But what you can see, the red cross means that the species had not been found. So basically, this is really a polar species, with the exception that many polar species you find actually in the Baltic Sea, which is not polar really, but it got, because it's so unsalted, it got frozen in the winter, and we find a lot of species actually from the Arctic in the Baffin. But interestingly, it was also found, we isolated from the ice, and it was also found in ice samples. The other interesting fact is that we had two species, one from the Beaufort Sea and one from Baffin Bay, which are a bit different environment. And the one from Baffin Bay, we never found the barcode anywhere. So there are these species that are such low concentration or so unique habitat that they are not found in the environment. So sometimes we say that we cannot culture what's in the environment, but sometimes we culture things that are not in the environment actually, but it's turned out to be a fragile species because we lost it. Okay, so another work which has been published last year. So this is a work from people from the University of Oslo, so the team of Bente Edvardsen, and also from the Alfred Wegener Institute, Uwe John. So the first author, Luca Supra, was a postdoc with Bente. And what he did is they did a cruise around Svalbard. So Svalbard is here, very north. It doesn't look very north, but it's very north. It past the polar circle, and there are a lot of fjords in Svalbard. And so they did cruise in different fjord, and they isolated a number of diatom species actually. So at the same time, they were also doing metabarcoding of each of these samples. So the question was whether the species that were isolated of Svalbard, which is a polar environment, are really all polar, or they are more widespread than really polar, and can we tell something about this? So the way, a bit of time, so I will be ahead of time actually. So what we did is what Luca did is a very rough distribution between different species. So there were some which are Arctic, so they are mostly, or you give a percentage, but they are mostly restricted. I don't remember exactly which percentage of sample you use, but which are mostly restricted to the above the Arctic sample, Arctic circle, not the sample. 66 north. Then some are Arctic temperate and some were cosmopolitan. So you find them almost everywhere. So I give you two examples. One is polar genotypes. So these are sequences which are exactly similar to this species that he isolated, Chetoceros diadema. What you can see is that you find it mostly only in the northernmost fjord, and the same for this one, Chetoceros convolutus. And what you can see on this map that again, when you have a cross, means that the sequence was not detected. You find again that really these are most of the sample where the sequence is found in the database are from Arctic location, from polar location, Arctic in this case. I think there is one here. It's probably. I'm not sure it's from your study or the one before, the same in Fielder's Bay, but it's found in the. Just with this, there is a very interesting question also to address, and that Nicole Lab is addressing whether some of the species which are polar are bipolar or only found in one of the poles. Some are purely Arctic and some are purely Antarctic. So this is still an open question because you find, based on V4, for example, some species that are found both. But maybe if you were using a more resolutive marker, you will see some differences. So anyway, and here is a cosmopolitan two species, Schonodiscus and Pseudonychia grani, and there you can find it in polar region, but also in other regions. So these were termed cosmopolitan. What's very interesting is that then you go back to just the metabarcoding data, and then you look at the sequence. So this is just for diatoms. You look at sequence that correspond to this three region, cosmopolitan, Arctic, temperate, or Arctic. These are these two locations are more towards oriented, receive a lot of Atlantic water, and this receive more Arctic water here, this fjord, which is north. What you can see is that at the Atlantic location, you have much more Arctic temperate species, while at this fjord here, you have an increase in Arctic. So there is clearly, a relationship between the strain or the genotype you find at a given location, and it seems, fairly obvious, but I don't think for plankton there are that many studies actually of this geographical distribution. Okay, and I'll finish with the enderized bloom, which was supposed to be, oops, okay, not touch anything here, where am I? Yeah. Okay, took. So, and I think Nicole introduced this this morning. These environments, polar environments, are very, very seasonal. You go from full sunlight to no sunlight. You go from open water to completely frozen water. And actually, in the slide she showed about the Antarctic, you can see that what the satellite sees is that there is a bloom around the edge of the continent. But actually, the bloom, and this has been shown only quite recently, although I'm probably a lot of work before it been done, for example, by Norwegian people. The bloom occurs, actually starts under the ice. That's the first stage of the bloom, and this was shown in a 212 paper by Kevin Arrigo, where it's a very short, it was in Science, but it was a very short, I think they stopped doing this Brevia paper now. You have to do a full paper or you are not allowed to publish in Science. So you can see here, for example, this is the sea ice concentration. So you can see that the sea is completely covered by ice and then no ice here. And you can see here this big, this is POC, so particle or carbon, correspond to phytoplankton, basically. And you can see this big bloom developing actually under the ice, and then when the ice breaks, actually the bloom begins to fade. So a large part now of the bloom in the Arctic is, and I think in Antarctic is probably the same, occurs first under the ice. The problem is very difficult to monitor. Of course, you cannot use satellite to monitor this, so you have to go there and do, I don't know how they did in their case, but in our case, so this led to this project called Green Edge, and which had two components. One was a cruise in Baffin Bay here. The other one was called Ice Camp. So what we did, I didn't do, but I participated. We made a tent on the ice, and then we made a hole in the ice, and then we had a CTD where we could sample the water column below the ice, and we stayed there 3 months until the ice melted. So we arrived in May, and we left late July. It was very critical at the end because you don't know when the ice melts, actually, because it begins to melt, and then it's more difficult. Like here, when I arrived, these pools were not there. I mean, it was just snow, and then the snow melts, and then the ice began to melt, and then we had really to rely on the Inuit, actually, because they told us, oh, tomorrow you have to leave, because then there will be no more ice, and they were, of course, very, very precise. So every 2 days we were taking samples, and we were monitoring a lot of different parameters. And what we were really interested in our case was the evolution of the ice algae and phytoplankton population under the ice. So the way the bloom develops, actually, there are several stages, and we kind of defined artificial stage by stage 1, stage 2, stage 3. This is basically, just look at this curve here. First look at this here. This is the ice thickness and the snow thickness. And actually, initially, there is no bloom under the ice because of the snow. So the snow stops really the light. It's not the ice that stops the light, it's the snow. And as the snow begin first to melt, then that's when the bloom is really developing. So this is the snow and this is the ice. So we were sampling just under the ice, so 2 different levels in the ice, and then a different level in the water. And as the bloom developed, we went deeper because the bloom was becoming deeper. What's really impressive when you have at the beginning is remember Osvaldo, when we were in the Pacific during Biosop, we were trying to beat this Secchi disk record and we were able to see a Secchi disk, so a small disk at 75 meters. And there it's the same, actually. It's incredibly clear, except it's not blue, it's more gray. But before the bloom, it's really very, very impressive. At 40 meters, you see the CTD like that. And then after it becomes, so anyway, so the water is initially very clear, and then it's developing by this underwater bloom. Okay. Don't remember what is the next slide, so I have to wait. Okay, so we were interested in doing the analysis of the plankton community, and this is work from Adriana's lab. So her lab is called Genomic and Ecology of Eukaryotic Plankton. It's a geek lab and at NTU, and this is work from Clarence with a PhD candidate with Adriana. So as you can see, so this is the ice here and the water, different level in the water, and this is a phytoplankton species. So this is very complex to analyze. It's a lot of things are happening, things are changing. You can see that there are some trends, but I'm not going to focus on this. I'm going to do just present you what using the biogeography, you can simplify a bit these patterns and make sense of them. So just to summarize this different stage, this is a different species, and you can see here it's quite complex evolution of species between the snow cover, the snow melt, and finally the ice melt. So to do this geographic, oops, okay. We did the same analysis that Luca did, but a bit more complex, and on the new version of Metabar 2, where we have about 6,000 samples. So you can see here all the samples that were in this analysis. And they are color depending on the latitude, whether they are polar, temperate, or tropical. And this is a full analysis of the database. Okay, so you can see that that's the location of the different samples where we find all these different species. And you can see, for example, this is a typical of cosmopolitan, but most of the ASVs we have are either from temperate, tropical, temperate, and or polar temperate. So this is different. Some groups like cosmopolitan, there are very few actually cosmopolitan species, and polar tropical, very few also, which makes sense. Of course. So anyway, so then we just looked at the sequence that we'll find during this underwater bloom, during greenage. And what we find, of course, is mostly, like in the case of Luca paper, polar or polar temperate. Okay, we have a couple of temperate and a couple of cosmopolitan sample ASVs, but most of them. So when we do this, so look. At the so here we have the ice, and here is the water. So two different levels in the ice, and four different levels in the water. I just want you to well, the first thing, okay, is that when you compare ice versus water, you have much more polar species, purely polar species in the ice than in the water, which makes sense because I mean, in temperate you have sometimes ice forming, but not so often. And also there is a case of the Baltic Sea, which bias a bit the analysis. But what's very interesting, just concentrate, so these three fractions, phytoplankton, less than 3 micron, 2 microns, nanoplankton, less than 20, and microplankton, larger than 20. So just concentrate on this here for the water, so the water and the phytoplankton. What you can see is that there is a very clear change. We are starting with alcohol species, but this is sequences or ASVs, which are dominated by polar here, especially at the beginning, just at the beginning when we arrived, where the water was super clear still, so no bloom yet, by polar species. And then as time goes, this becomes completely dominated by polar temperate. What you can see for the nano and macro, it's more stable actually, but micro is quite striking. So some example of polar species is that the species that you find in the first stage and second stage, like Ankyloprises, for example, Baffinella. No, this is Baffinella. This is Ankyloprises. Sorry, Ankyloprises is here. Baffinella is here. Or Triparma. Triparma is a very interesting group, which is very small cells, very close to diatom phylogenetically, and that have silica shell actually. So they are found in quite few polar environments. And then you have polar temperate species, and these are species that are when global change will occur or takes place, when you have will probably becomes much more important actually in these environments. One is Assistis. So here, so you have two stages. You have this stage, which is a flagellate stage, and this stage, which is colonial. It's not to scale, and this is much bigger actually. This is very big colonies. And actually, when we were in Baffin Bay, they were seen from the boat and from sampling, and people were saying that it was very unusual actually. So it seems like the species that's going to be more important in the Arctic were coming. And as the same as Micromonas polaris, which is a very small green algae, which is about 1 to 2 micron, and which has already, there was a paper by Bill Lee like 10 or 15 years ago that was saying that phytoplankton was increasing in the Arctic, and it was probably due to the species, because this is the only real phytoplankton species in Arctic waters. The other one are more nanoplankton actually, Baffinella, Phagotrophs, and so forth. So, so these species are the primary culprit if change occurs. So they are very important to monitor actually. Okay, so I summarize here, saying that all these metabarcode data are very useful, but they need to be in a shape or form that can be used. So that was a purpose. This is the purpose of MetaPR2. And if we look at, especially in polar systems, it seems that phytoplankton and more generally protists have clear biogeographical distribution. Now, what we did is just looking at latitudinal, but it's of course not enough. We should look at the temperature range, but this is more of a problem because for many of the samples that we have, we don't have the temperature. People didn't record the temperature, or it's very difficult. So some studies have temperature, but some others don't. So, so that's one of the perspective actually. One of the perspective is on this database is to add more public data sets, and there are at least more than 500 published data sets. Some are interesting, some are not interesting. What's especially interesting will be have more data set from the Antarctic, because we have some data set, for example, from the peninsula, but the peninsula is it's not even polar. It's above the polar circle, right? So it's but we need more data especially to compare with the Arctic. But also from other environments like soils, also microbiomes. People are now studying protists on microbiomes, not only bacteria. What will be really interesting is to have also associated with the samples some more metadata, and also for the species like the trophic mode, the size class, and also, for example, look at the temperature range. But these are very difficult studies, I mean, to do, for example, temperature range for even a culture. It takes a lot of time, and people don't want to do this now. Also interactions, networks, like what Matt was showing. And finally, there is also the advent of long-read sequencing, which will be very useful to get for the full S, 18S, but also the ITS to get more resolution, taxonomically especially resolution. Okay, just some people to thank. First, all these people that have been reading, doing Metabarcode paper because they are very useful. Some projects like Biomarks, Ukrave, yeah, and also CNRS, Sorbonne University, and Nanyang Technological University. And finally, all the people from the PR2 team, because it's a very big effort. The PR2 has been fed by specialists of different groups. Right now, I'm working on the project with Javier del Campo from Barcelona, but also with a scientific committee that helps to try to find money, define direction, and so forth. Metabar 2 has been done mostly with Adriana's lab in Singapore, some of her PhD students and undergrad, and also with the lab of Fabien Burki and Mawash Jami. And finally, there is another small database called PR2 Primers, which I didn't mention. And finally, I want to thank the team of Adriana, the geek team in Singapore, very nice team, and which has been really very, very supportive of all this work. And this is Clarence. Adriana is here, and Clarence, a PhD student working on Greenwich. Thank you very much, and we'll be ready for questions. If there is time. Good.
Thank you, Daniel. I'm sorry about the technical issues.
No, c'est bon. I didn't panic. I didn't.
Okay.
Didn't throw the computer.
Okay.
So we have a couple of questions.
Hello, I'm so gratefully of this symposium and the information of your presentation. Thanks. My question is if metagenomics can be used as a reservoir of information and biodiversity of microbes, thinking about the future and actual consequence of the climatic change can produce in the communities of microorganisms.
Okay, so first thing, it's not metagenomic. It's metabarcoding and not metagenomic. Metagenomic is when you look at the whole sequence of the whole community and without targeting a specific gene. I'm making this remark because a lot of people are mixing the two, and I think vocabulary can be sometimes important. I think it can provide a lot information for first on the evolution of a system. So there has been some studies. For example, there was one in Barcelona where people have been monitoring for 10 years. And there is one in Rosco, actually, who have been monitoring now for more than I think we started in 2005, and so it's almost 15, 20 years, and we keep doing it. So I think this metabarcoding, especially because these methods are very well fixed now. They are. I mean, there are differences in extraction, of course, in sampling strategy, but more or less it's quite stable. So it's not something that you have to change every. It's like there is a. In comparison is with what's called the Plankton recorder, which was sampling done from the Plymouth lab since the, I don't know if it's from the 30s or from the 50s, but anyway, it's an incredible database of plankton that has allowed actually very high impact papers to show that there are intrusion. I mean, this what's called the Atlantification of the Arctic Ocean actually. So there are species, and I think we can see that. Whether we can predict now, this has to be done in conjunction with modelers, of course, because people need to model. So there are, for example, Micromonas, the species. There has been some papers where they have been doing some analysis of the temperature range, response of growth to different temperature, and that then try to predict how the species will evolve as a function of different scenarios and so forth.
Any more questions?
Yeah.
Following on your modeling comment, Demi and Abelard has the new niche modeling.
Sorry?
Demi and Abelard has that new niche modeling where he looks at biogeographic distributions and then estimates. Have you heard of niche modeling where?
Yeah. Okay. Yeah.
And so it's pretty new, but it takes the biogeographic data and suggests what the actual niche would look like for each organism. It seems like what you're doing creates. He hasn't tested it with microbial eukaryotes, but it works really well with like prokaryotes.
Yeah. No, that's. I think also there is a lot of things with AI. I mean, all these methods with AI where you could use this. No, it's the problem of all this database is to get the traits associated. Especially, and you can get metadata on the temperature, but even during Tara, there are some samples where you don't have the data, so you have to use like average data or things like this. So this is a bit less precise already. One trait, for example, that we want to incorporate into PR2 and then translate to MetaPR2 is the whether they're autotroph, heterotroph, phagotroph, because we say phytoplankton, but they are probably phagotroph. And phagotrophy is very complex because the species can be sometimes autotroph, sometimes phagotroph in the environment. I mean, if it has the capacity of being phagotroph, it's not phagotroph all the time. So this, I think that's. But I think my feeling is that there need to be more data associated with the organisms themselves. So get more data on, what you got with long time ago with Prochlorococcus. I mean, all these studies where you can define precisely the range of but these are very tedious studies that people don't want to do, but they are needed actually.
Yeah, what I was thinking of was you could use the model and your new biogeographic database to make predictions about what those traits should be. And then, the physiologist would have a very ecologically contextualized targeted experiments that they could do. And that would be exciting to see that interplay between the physiology and the modeling and the biogeographic effort you've done in the database.
No, definitely. I'm part of it's a project a Norwegian project that's brewing, where they want actually to go back to many data and that they have, because there are many data sets they have, and put them all together and do this kind of prediction, and then after do some kind of experiments. Actually, Luca, who was the author in the diatom paper, he had a proposal to do this, but it didn't get funded. It's not such so sexy actually, which is bad, because we have the data. He was proposing actually to do a temperature, this battery test with temperature, and which I think it's very useful actually, but yeah. Nicole, hi. Technical question.
A little bit. Nice talk, Daniel. Thank you very much. I have a question about the unquantitative issue of the metabarcoding with eukaryotes, because we know that most of the difficulties came from the amount of, or the number of operon for 18S,
Yeah.
gene that we do not know? And do you think that we can like include in this global database information for the groups that we have that are not so much, information about the copy number of the ribosomal operon or something else, so we can standardize and make more quantitative numbers out of
No,
metabarcodes?
that's a very good question and an active. I know of some groups that want to try to get these estimates through from the metagenomes actually. By comparing like using some reference gene, which is single copy, and then comparing with. The problem is that if I'm not so familiar with metagenome analysis for eukaryotes, but I think the etiness is undersampled in the genomic data. So, but if it was well sampled, actually you could do some kind of estimates, and then comparing this to studies like Tara, where they have also cell count. So I think we can, I think, reduce or get some boundaries on this bias now. Yeah, because, but I still think that actually having like macroscopy data are critical to try to compare. So that's a trick. For many species also, especially the larger species, we don't know the number of copies, right? So like dinoflagellate, we have some data, but and it's not very difficult to get actually this number of copies. I mean, we did this long time ago with Zoo, and by qPCR you can get this. So that's a problem. I think everybody is doing metabarcoding because it's so easy, but there are not that many studies actually that are before metabarcoding and try to add some more information about how quantitative it is. But I agree with you. It's
Thank you very much.
Any more questions?
Okay, thank you, Daniel.
okay.